All right, so I've been asked to provide a bit of an overview of Agriculture and Agri-Food Canada's approach to soil organic carbon measurements. And I want to say straight out that we don't have a standard measurement approach across all the living labs. You will see that there are 13 living labs being established or already established across Canada. And there's some consistency amongst the different living labs, but lots of living labs are taking different approaches. So what I'm going to be describing here is a plan that we've come up with working with Shabtai and working with two really fantastic colleagues, Ben Eller in Lethbridge and Louis-Pierre Como all the way out on the East Coast. So this is something we've been kind of bouncing around that we think would work relatively well for whoever might be doing the measurements in individual living labs. It's consistent with guidance that Ben has produced in the past and pretty consistent with international protocols and would be pretty easy to implement. But that being said, this isn't hard and fast rules. And so the work that we're going to be doing over the next 2 days in this data protocol workshop will be really valuable, I think, to help refine those and justify any adjustments that we make. Another reason why I wanted to bring up this slide is because the Agricultural Climate Solutions Program has three main stated goals, and that is number one, to sequester carbon, number two, to mitigate greenhouse gas emissions, and number three, to help promote co-benefits that might be associated with management practices that are adopted by participating farmers. But that first one at the top, sequestering carbon, really bugs me because I don't believe that sequestering carbon is a good goal. I think we want to increase soil carbon perhaps, but sequestering carbon to me is not possible and not a wise move. Healthy soils cycle carbon. So what we really want to be doing is increasing the net amount of carbon going in more than the amount of carbon that's coming out as decay. So I just want to put that out there right now that I really don't like that word, sequester carbon. Let's talk about maybe increasing soil organic carbon by managing the flow of organic carbon through the soil. So that's a big piece of emphasis that I think is important, and my colleagues Ben and Louis asked me to specifically make sure that we make that point. Okay. All right, so if we're talking about measuring stocks or the content, the amount of carbon in the soil, we've divided it into 4 main steps. And I'm leaning heavily on this publication that several AAFC colleagues, Ben Ellert, Henry Jansen, Bert Vandenbeigart, and another colleague, Eric Bremer, have put together. But you'll see a lot of these steps echoed in many protocols that have been published. So we're going to divide it into a few steps: sample collection, extracting soil samples from cores, processing and analyzing the samples, calculating soil organic carbon, and then repeating it all again in year 5 of the project and hopefully beyond. Oh, yes. And another thing that Louis asked me to make as a point is that these protocols that we're mentioning here, or this guidance that we're describing here, might be appropriate for soil organic carbon stocks, and you could use those samples to do some measurements of soil health, for example, but you may want to sample things and process soils differently for certain measures of for other parameters of soil health beyond soil organic carbon. So we're really focused on what we might want to do for soil organic carbon, but not necessarily say for, I don't know, nematodes, or Tom Forge will be here soon, so he will speak up about that. Okay, so what I've done is divide these 4 stages into the ideal guidance that you're going to see in a protocol and then some practical problems that always we will encounter, and some tentative solutions to dealing with those problems. So number one, prior to our visit, we should get a bit more information about the site. I think this is really appropriate when we're talking about living labs, because we're already talking to farmers a lot. We have a good sense of their farm, the kind of practices that they already use, and the kind of practices that they like to experiment or adopt as part of the living lab. And we want to understand, some of the challenges that they're facing in terms of coarse fragment content, slope, and we want to consider those kind of factors when we're developing our sample collection approach. We want to walk around the site, and if possible, try to identify some areas that are relatively uniform. If it's available, you'll see this a lot. If there's prior information available on soil organic carbon, we can use that information to calculate the number of samples that are required to effectively characterize the soil organic carbon content and calculate a minimum detectable difference if we're wanting to compare, like a control and a treated area. And we also want to think about what might be an appropriate sampling pattern. And this is going to really vary depending on the farmer and the size of the field that is available and the growing system that they're using. Some living labs are taking an approach that is using a grid sampling pattern. Others in the Peace Region Living Lab, they're deciding, once they've designated a relatively uniform area, that they're going to sample in a W pattern, and to make sure that it's spread, in a representative way across the sampling area. That can be determined. Oh, and I should have mentioned at the bottom, I'm not talking here about, designating a control area and a EMP area. Let's just talk about, sampling one area to characterize it. And then once we know where we want to collect those samples, we need to record the GPS locations of each of those sample locations so that we can revisit those on our resampling day. And we know exactly where the samples were taken. This is also important because for many living labs, other parameters are being measured at those sample sites. For example, residue or additional samples are being collected for aggregate stability or nematode populations or yield. And so we want to try and be able to relate the soil organic carbon measurement that was taken at that spot with other variables that might be collected. But then there's reality. So guaranteed, I work a lot in orchards and vineyard systems in these tiny little farms. They're often on a slope. There's often one soil type over here, and halfway through there's an old riverbed, and you are going to probably, in many cases there, need to consider stratifying the area to accommodate those differences and to make sure that you're collecting representative samples from what you're getting within that field. So you can consider stratifying your sampling plan to accommodate those differences, for example, based on slope position. And then this is probably going to be the case a lot, that we don't have pre-existing soil organic carbon data already, or if there are, the number of samples that minimum detectable difference calculation tells you to do is hundreds. So that's obviously not practical, and you may end up with no soil left if you do that. So we've decided that let's aim for 9 or 10 samples within our ½ hectare to 1 hectare area to try and capture that variability. And that's the approach Paresh Fresta, who introduced himself online, is taking. He's collecting samples in this W-shaped pattern across each representative area, and he's aiming for 9 samples within that area. And then we have a problem that I see a lot. You can see that with the picture on the top right, that there are really obvious differences imposed by your management practices that you also need to accommodate in your sampling. In many sampling protocols that I've measured, there's no mention of this kind of thing, but when we walk around on the ground, we can see those. It's very obvious that the drive row vegetation in that orchard is going to have driven very different soil organic carbon contents than the crop row, which has been left bare because of applications of herbicide or tillage. So we need to make sure that we collect samples from both those types of soils. All right, and then we get down to sample extraction. Some of you in this room have been hearing for a year and a half about my and Shabtai's mission to get truck-mounted hydraulic soil corers. We are still frustrated in this mission, but this is the guidance that we have been told, and damn it, we're going to get them. So we've been asked to make sure that we use truck-mounted hydraulic soil corers with a soil corer tube with an internal diameter of about 7 centimeters. This diameter is being recommended so that we can get sufficient sample to do measurements of bulk density and soil organic carbon on the same sample. This is the ideal situation.
How much do those cost?
Oh, Shabtai, how much do those corers cost? Can you remember? Shabtai's dealing with the purchasing.
Yeah, I mean, there's various shapes and sizes and models, but the one we're trying to get is about 50,000 Canadian. It might be a little bit less than that, but we budgeted for about 50,000 Canadian, and that will give you a number of tubes and the mounting equipment that you need and so on. Smaller ones are cheaper.
That's another ideal situation, is you have 50,000 hanging around. Okay, then again, in the ideal situation, we would drill these cores down to 100 centimeters and in the field, break these samples into distinct soil depth segments. So we collect the surface litter material, and then we collect samples, separate samples into sample bags of 0 to 15, 15 to 30, 30 to 60 centimeters, and then 60 to, if we can get it, 100 centimeters. In the field, carefully placed in the labeled sample bag so we don't lose any material. And then, like I mentioned, we can use those samples both to determine soil organic carbon and bulk density on the same sample. Again, this is the ideal. But in reality, the site might not be accessible by the truck or tractor to which you have your hydraulic corer sampler mounted. And this is going to be the case for many of us. Coarse fragments may prevent the use of the hydraulic soil sampler in an effective way, or maybe the soil structure is so loose, maybe the soil is really wet or dry, or it's very sandy, and so we can't use that hydraulic soil sampler effectively and preserve the soil structure. So in that case, you'll see, where's Hannah? Hannah, that is Naomi Yamaoka collecting that sample up in the right-hand corner there. And that is often the reality, is that in many cases, I think we're gonna have to sample by hand. And then just collect very careful soil samples for bulk density separately.
We have a question from us.
Yes.
They say that the soil corner will not fit in the grade rows. So
Yeah, so Hans will be happy to know that Medi has a tractor mounted hydraulic soil sampler that has been designed specifically to swivel into the vine row. So we do have that soil sampler available, and we've been practicing with it at the research station. So we do have that one accessible. I'm so glad Medi saw into the future and purchased that several years ago. So we've got that, and we can mount it on a truck or a tractor. So that's a good one. Another problem that often happens is that the soil will fall out of the 7-centimeter corer. It's quite a surprisingly large diameter to try and keep the soil intact. So in some cases, we can move to a smaller soil corer. And in fact, we have a slightly smaller soil corer available for that tractor mounted sampler that I just mentioned that my colleague Betty has. So that can help solve the problem. And another issue we have is if we're collecting samples down to 100 centimeters and we're dividing them into these different segments, and the top 2 segments are only 15 centimeters, in some cases, there's so many analyses that we want to do with that sample beyond organic carbon. We just don't have enough sample for those top 2 quite shallow depth increments. So in that case, and this is a problem that, again, my colleague Bharat Shrestha has encountered, that he's had to go and collect some additional samples from those shallower depths, just to make sure he has enough sample for all the other accompanying measurements that he needs to do. And then we get back into the lab and we have sample processing and analysis. So in the ideal situation, we dry the samples, we grind them, we sieve them to 2 millimeters, and they're ready for analysis. So we have this nice process sample ready to go. And in order to make sure that the aliquot that we pull out for analysis is representative, many of you here will have looked at a pile of dry soil on the counter, and you'll see all these sand particles kind of like falling off to the sides, which means it's very difficult to get a representative sample that accurately reflects the texture of the sample. So I have a riffle box in my lab. Other people use centrifugal samplers. That's what Ben Ellert swears by. And that can help you get a representative aliquot for sample analysis. And then you determine soil organic carbon using dry combustion, like ALECO, which Shabtai and I have in our labs, and determine total nitrogen at the same time. We recommend this because for 2 reasons. Total nitrogen is a really useful measurement. Hi, Tom. And also, when you look at your data, if you have a super high carbon to nitrogen ratio in your final number, that can be just a sign that there's something weird with the sample, and you might want to go back and take a look and make sure that either there's not a huge amount of inorganic carbon in there, or you didn't just sample a chunk of wood or something like that. So it's just useful to do those measurements simultaneously. But again, then we have reality. And in many, many cases, we are going to encounter really coarse roots like you would see when you're sampling at a big old apple tree. You're going to have coarse fragments in there, stones and cobbles potentially, and these will interfere with your proper bulk density measurements. So when you're sieving, you need to pull those stones and roots out and weigh them. Lots of us don't have riffle boxes or centrifugal samplers. And so I know what a lot of people do is this cone and section method, where you just pile your sample onto the counter and then you just cut out a little pie slice. And that way, any, sand that's rolling off the top of the side and pooling at the base of your little hill of soil, you'll also get that sample those materials when you're collecting your aliquot. So that's another approach if you don't have one of those fancy other pieces of equipment. And this is a big topic of discussion for me and for Ben Ellert in Lethbridge, is that we often have samples that have a significant fraction of inorganic carbon in them. Many of the people on the coast are probably used to doing measurements of total carbon in their soil and being able to confidently assume that all that carbon is organic. But when you have soils with a high pH, you can't necessarily safely assume that. So we've decided to take an approach that colleagues in several other countries that I've spoken to have recommended, that when they have their samples ready to analyze, they first measure pH. And if the pH is greater than about 6.4 or 6.5, then they shunt that sample off into a different pipeline where they analyze it for both organic and inorganic carbon. And there are many approaches that you can take to do that. The most common is acidification, where you have an aliquot where you measure total carbon, and then you have another aliquot which you first acidify and then measure total carbon. And the difference in those two is the inorganic C. So you can calculate organic and inorganic C just by difference. Another approach is to do a two-stage combustion method, where instead of acidifying to get rid of the inorganic carbon, first you muffle or burn the sample at like 500 degrees Celsius to get rid of the organic carbon, and again measure carbon in a non-muffled and a muffled sample and measure the two different fractions by difference. Ben Allard is sending me a calcimeter next week, so I'm going to experiment with using that method to calculate inorganic C. I have no experience with it, but we'll give it a try. So there are a few different approaches that you can take. Yes, great. Okay, and finally, soil organic carbon stock calculations. So in order to calculate soil organic carbon content for an area in kilograms of carbon per hectare, we need to have that bulk density measurement, and then we need the concentration of organic carbon for each depth increment. And then with a few little neat changes to make sure all the units match, we can get soil organic carbon in kilograms per hectare. And we can sum them for each depth increment down to this total depth that we sampled to get a total content measurement. Again, that's the ideal. But and there's a huge variety of approaches to measuring soil organic carbon for content. For a very simple measurement, there's a lot of disagreement about the proper approach. But I think we're moving to a consensus where the best measure is essentially the organic carbon in the fine fraction. So the material that passes that 2 millimeter sieve divided by the volume of the total sample. So that's the approach we're recommending. And another interesting complication, this is something that Sean's brought up several times, and certainly Ben Ellert would support it, that it's not sufficient just to accommodate those bulk density differences, but we also have to take an equivalent soil mass approach. And the reason for this is shown in two figures down here. So the figure on the left is given to me by Ben Ellert, showing that when you till, you change the bulk density, and then if you're collecting samples to the same depth, you're actually sampling a different kind of subset of the soil. And the figure on the right is from a paper that just came out by Fowler. I haven't read the whole paper, but I thought the figure was great. Again, showing that if we change the bulk density, so say we increase the organic matter of the soil, which is exactly the aim of adopting the management practices that we're investigating with the Living Lab, then when we come back and resample, we may be sampling a slightly different subset of the soil. So we have to correct for that using this equivalent soil mass approach. Okay, the last couple slides. So we've done all those samples in year one, and now we need to come back several years later and see if the adoption of our management practices has done what we were hoping to do. Did we actually increase soil organic carbon by using cover cropping, for example. So it's been suggested several times that maybe it would save money if we just measure an area at time 1 and then come back in year 5, measure the same site, and voila, we can see our changes. But this figure I wanted to include to show that we actually need to measure our control area and our management practices area both in years 1 and year 5, because what happens sometimes this soil organic carbon kind of goes like this in both areas. I know this happened with colleagues who were working in Saskatchewan, that they found one year they went back to remeasure and soil organic carbon was really low in both treatments because of a massive drought that they'd suffered for a couple of years. So if they hadn't had a control and a treated area and just measured the treated area before and after, they would have accidentally concluded, oh my God, like our no-till treatment has reduced carbon. But because they had both, they could see that the differences were actually increased with time. It's just that they were both lower. So this is an important thing to keep in mind. And finally, this is another figure taken from Ben Ellert's protocol. And I wouldn't pay attention too much to the distances between these little sample points in this figure, but I just wanted to make the point that this comes right back to why we recommend taking the GPS locations at time 0, is so that we can take measurements quite close to where we took our original measurements, just so we're making sure that we're measuring the same things. That being said, we don't want to take them like right next to each other, because we could accidentally sample exactly where we were before. And also sometimes what happens is you leave this nice big hole, and then over time, the surface material falls into the hole. So you're going to end up with this hole with massive, gorgeous, organic carbon rich core, but just like this little carrot of beautiful soil with a desert around. So it's good to take it, a good meter or so away if possible, if it's still representative. So sample close, but not right where you sampled before. Okay, so in case, I don't know if we want to, we'll probably be able to share these slides.
Absolutely.
Okay, great. So I just threw up some of the slides, some of the references that I mentioned here. There are so many awesome protocols out there, and I'm sure there are lots of things that we could improve in what we've laid out here. So don't use these as the Bible, but these are just a little start. And I thought that this little YouTube presentation from Jonathan Sanderman is so great. So I totally recommend that one. And with that, I'm looking forward to hearing about Sean's suggestions and having a discussion.
Are there any clarifying questions for Kirsten now? We could take a couple of questions. Yes, go ahead.
I have 2 questions.
Yeah.
One is about a number of samples. So and just clarify, we have a 1 hectare treatment site and 1 control site, is 1 hectare. So we want 10 samples, which would be 10 pores in that 1 hectare, and you want samples on 4 different levels. So that's actually 40 soil samples we have to send to the lab for control. So that would be on each farm site, 80 samples.
Yeah.
And we could, okay, I got that number. How much we got to send in? I'm just looking at our budgeting.
Yeah.
So that's about 80 samples. Okay. Per farm. Now, my second question is on all the analysis for the soil organic carbon. Can we have the lab do all that work for us?
Why don't I I'll go up to the front so that,
Okay.
yeah. So, okay. This is great, Dave. I'm glad you asked this question. So we at AAFC have budget to do the soil organic carbon analyses. So each commodity group participating in the Living Lab kind of wants different support from AAFC to collect, process, and analyze those samples. Some of our colleagues, like with cattle forage, they'd like to do the sample collection and processing themselves and are going to send us the samples to analyze. And others have asked us to come with our hydraulic soil sampler and collect the samples and help get our students to process them and analyze them. So that's something we can work with you in vegetables to figure out is the best, how we can best get that done.
Just to follow up, so we've been doing lots of soil samplings in the Delta with the data growers. We'd always send our samples to the lab, ensuring that they would do the whole thing, nutrient value and organic matter and all that. Is that process okay?
Yeah, I mean, that process is okay for, it depends what you're using it for. So are you talking about for soil organic carbon? Yes, so we can use those numbers and take a look and see what they suggest you're seeing. But if we can get some samples, particularly in the first and last year of the project, collected in a way like I described, it would be great, just so all our data from all the commodity groups speaks to each other in a similar way. But we can come and work with you on that, Dave. I know I've spoken to Sean about coordinating a bit with him, and Shabtai and I have talked about talking to you and Lisa, and maybe we can do some sampling this fall if it works for everybody.
Yeah, and we can have a chance to get more into the specifics once we have a chance to like break out in the discussion.
I don't belabor it, but we gotta get answered because we're gonna be starting on Monday on one of our farm sites.
Right.
You probably heard that that's that site that's big controversy in Surrey that maybe might go on agriculture, and they're the earliest native ground in British Columbia, and they're ready to prepare. So we want to take the soil sample before we bring in this compost. So we're going to go ahead and do it this spring and then share the information with you and
Yeah,
maybe adjust it for next year or whatever.
that sounds great.
Okay, thank you.
And Jeff, did you want to ask one question?
Well, so I'm just trying to get my head around, you're talking about a W pattern
Mhm,
on a hectare.
mhm.
So if I have a control area,
Yeah.
okay, I have a control area and a treatment area, and in dairy, those control areas and treatment areas probably will be 4 hectares, 10 acres. So you're gonna do a W pattern, 10 samples in the W
Mhm.
on each.
9 or 10, yeah.
Okay.
Yeah.
I just, if we're gonna be talking to our drawers about access,
Right,
that's a lot of disturbance.
yeah.
And the
Okay.
timing will be, yeah.
I didn't put this in the parking lot,
Yeah.
so it's like something that we know that. it
Yeah,
probably
I just-
should be-
Just talk about
Yeah.
Yeah,
Great.
come up with protocols for coordinating and timing, because that's not something I mentioned, but we'll want to make sure that the timing each year when we do it is similar relative to, you know, farming activities. So, but yeah, we just need to figure out the protocol and best timing for everybody.
we're gonna make it easy for you. We're gonna get those sites that have no forest fragments. But just one more question about the pH. So that's native pH.
Mhm.
What if it's been modified by liming and is over 6.5?
That's okay, because the lime's inorganic feed, so we need to account
So you
for
would
it.
have to do that.
Yeah.
calculation. Okay.
Yes, John.
This is someone who limited soil sciences a long time ago. I'm just curious about what the relationship between pH and inorganic carbon. Why is high pH having more than that?
Yeah, well, inorganic C is something I've had to learn about reluctantly. we don't see it a lot in forestry, and I have some crazy stories about carbon isotopes that made me realize I need to understand inorganic carbon. But in the Okanagan, there's not a lot of precipitation. And because of that, cations accumulate in the soil, and through natural processes, roots create high carbon dioxide concentrations in the soil, which forms carbonic acid. And this carbonic acid will bond with the calcium or magnesium ions that are just hanging around in the soil because they haven't been washed down in the semi-arid climate because it's too dry. And so we get these accumulations of the calcium that's hanging around in the soil and this carbonate that was formed by root activity in the soil. So this is only really possible when the climate is quite dry and the soils don't get leached out regularly, which is different than in the coast. And so these inorganic carbon accumulate. And an indicator of that is the pH, because hydrogen ions will be leached out with the calcium, driving the pH down. So in our case, in the Okanagan, we get this accumulation of inorganic carbon, which is why we have quite high pH. Another cause of high pH is parent material. So if we have like limestone or there's all kinds of calcium rich rocks that can also form soil, and in those cases the inorganic carbon can be really high. There's only patches that I'm aware of in the southern interior. So again, we just need to account for those. It's like fossil inorganic carbon that can mess with our measurements.